Jambo Liam in for bear. Why does a metabolic enzyme mutation produce an epigenetic block to differentiation? Can you explain it, bear?
A bone marrow biopsy. The marrow is packed with blasts, immature, arrested cells that cannot grow up. The diagnosis: acute myeloid leukemia. The patient's sequencing report comes back. One amino acid change in a metabolic enzyme called IDH-1. That enzyme normally runs a step in the citric acid cycle. Nothing to do with cell division, nothing to do with growing up. And yet somehow, that single amino acid change froze the entire leukemic population in an immature state. A metabolic enzyme mutation should change how the cell extracts energy from nutrients, not lock cells into an immature state. Here, a single amino acid change in IDH-1 causes cancer. Why does a metabolic enzyme mutation produce an epigenetic block to differentiation? The naive picture is straightforward. Cells differentiate by reading developmental programs, genes that switch on in sequence as a blast matures into a functional neutrophil. The cell's metabolic machinery and its developmental programs seem to be completely separate systems. Change a metabolic enzyme and you'd expect to change metabolism. Full stop. But the genome's gene expression machinery needs cofactors to work. Methyl marks that silence genes are written and erased by enzymes, and those enzymes depend on small molecules from metabolism to run. The metabolic state sets the cofactor supply, and cofactor supply shapes the epigenome. Here is what IDH-1 normally does. It converts isocitrate to alpha-ketoglutarate, a metabolite that the demethylase enzymes need to remove methyl marks from DNA and histones. When methyl marks silence a differentiation gene, the demethylases use alpha-ketoglutarate to strip the mark and restore transcription. The mutant IDH-1 enzyme carries one amino acid change in its active site. That one change corrupts its chemistry. Instead of making alpha-ketoglutarate, the mutant enzyme runs a different reaction and makes a structurally similar molecule called 2-hydroxyglutarate, an oncometabolite that the cell does not normally produce. 2-hydroxyglutarate is the structural mimic. Its shape is close enough to alpha-ketoglutarate that it fits into the demethylase active site, the same pocket where alpha-Kg would normally bind and power the reaction. But 2-HG cannot complete the reaction. It sits in the active site like a key that fits the lock but does not turn it, blocking the real key from entering. With the demethylases competitively poisoned, methyl marks accumulate across the genome, including on the promoters of the very genes that direct blast differentiation. Those genes go silent. The blasts cannot read their own maturation instructions. They are stuck. IDH mutations appear in roughly 10% of acute myeloid leukemia cases and in most low-grade gliomas. In the leukemia, the result is a marrow overrun by blasts that pile up without ever maturing. It is a cancer of arrested development. Cells that are immature not because they are proliferating too fast, but because they cannot read the next instruction. Consider one IDH-1 mutant AML patient starting Ivosidenib, a drug that blocks the mutant IDH-1 enzyme itself. Day 0. 2-HG is 1,000 times normal. Demethylases are 90% inhibited. Blasts are clogging the marrow. These numbers are illustrative. Day 14. Ivosidenib has blocked the mutant enzyme. 2-HG has halved. The demethylases are recovering. Day 28. 2-HG is near normal. The demethylases are active again, and on the blood smear the blasts are maturing, acquiring the segmented nuclei of functional neutrophils. The cancer resolves not because the cells died, but because they finally grew up. A metabolic enzyme mutation changes energy metabolism, and, through the metabolite it floods the cell with, also poisons the molecular erasers that clear silencing marks from differentiation genes. The answer to the question, 2-hydroxyglutarate, is a structural mimic of the cofactor the demethylases need. It fits their active site but blocks the reaction, locking methyl marks in place and locking the cell in an immature state.
Here is what the body demonstrated. IDH-1 carries a neomorphic mutation. It does not just lose its normal function, it gains a new one. The mutant enzyme produces 2-hydroxyglutarate, an oncometabolite that poisons by resemblance. It fits the active site of alpha-Kg-dependent demethylases but cannot complete the reaction. Competitive inhibition. The erasers stall. Methyl marks pile up on differentiation gene promoters. The blasts are stuck not because they are dividing too fast, but because they cannot read the next instruction. Ivosidenib removes the source of 2-HG, block the mutant enzyme, restore alpha-Kg supply, and the differentiation program restarts. The trade-off: this only works in IDH-mutant disease. The mechanism is the therapeutic window. Take this prompt, run it on your own, pick any cancer type or clinical scenario you know about, and ask how this mechanism applies there.